Genetic engineering represents one of the most powerful technologies in modern biology, enabling scientists to transfer genes between completely unrelated organisms. In this video, we'll look at what genetic engineering is, how scientists transfer genes between organisms, real-world applications, and benefits and concerns of genetic engineering. Let's start by defining genetic engineering and its unique capabilities. Genetic engineering involves extracting genes responsible for desirable characteristics from one organism and inserting them into another organism's genome. This process transfers specific traits directly between species that could never naturally interbreed. Unlike selective breeding, which requires many generations of controlled reproduction to enhance existing traits, genetic engineering achieves precise modifications immediately. Scientists can add characteristics from bacteria to plants or from humans to bacteria, combinations impossible through conventional breeding. The genetic engineering process follows a systematic sequence of carefully controlled steps. First, scientists identify and isolate the specific gene responsible for the desired characteristic using restriction enzymes. Restriction enzymes function as molecular scissors, cutting DNA at precise locations to extract the target gene. These enzymes recognize specific DNA sequences, ensuring accurate removal of the desired genetic material. The isolated gene must then be inserted into a vector, a biological vehicle for gene delivery. Common vectors include bacterial plasmids, which are small circular DNA molecules, or modified viruses that can penetrate cells efficiently. Different organisms require different vectors for successful gene transfer. Plant cells might receive genes through bacterial vectors, whilst animal cells often require viral vectors designed to integrate with mammalian DNA. The vector carries the gene into target cells like a molecular delivery system. Once inside target cells, the new gene integrates into the organism's genome, becoming part of its genetic blueprint. If modification occurs early in development, such as in embryos, the entire organism develops with a new characteristic. Successful integration means the organism's cells produce proteins coded by the inserted gene. These proteins create the desired characteristics, whether insulin production in bacteria or pest resistance in crops. Genetic engineering has produced remarkable medical breakthroughs, particularly in pharmaceutical production. Bacteria modified with human insulin genes now produce vast quantities of this essential hormone for diabetics. This bacterial insulin is identical to human insulin, eliminating previous reliance on animal sources. Before genetic engineering, diabetics depended on insulin extracted from pig pancreases, which sometimes caused allergic reactions. Now, genetically modified bacteria produce pure human insulin safely and efficiently in controlled fermentation tanks. Scientists have engineered bacteria to manufacture numerous therapeutic proteins, including human growth hormone for treating dwarfism and clotting factors for hemophilia patients. These medicines would be impossible to produce affordably without genetic modification. Agricultural applications have created crops with enhanced characteristics benefiting both farmers and consumers. Genetically modified plants can resist diseases, tolerate herbicides, and repel insect pests without chemical pesticides. Some corn varieties produce bacterial proteins toxic to caterpillars but harmless to humans. This built-in pest resistance reduces crop losses and decreases pesticide use, potentially benefiting the environment. Gene therapy represents genetic engineering's most ambitious medical application. Researchers are developing treatments that insert functional genes into patients with genetic disorders. This approach could potentially cure conditions like cystic fibrosis by replacing faulty genes with working versions. Despite these benefits, genetic engineering generates significant controversy and legitimate concerns. Critics worry about unforeseen long term consequences that might only emerge after widespread adoption. Environmental concerns focus on genetically modified crops potentially affecting ecosystems. Modified plants might crossbreed with wild relatives, spreading engineered traits unpredictably through natural populations. Reduced biodiversity represents another ecological concern. GM crops might harm beneficial insects or reduce wildflower populations on farmland. These changes could cascade through food webs, affecting birds and other wildlife dependent on agricultural ecosystems. Consumer concerns about GM foods persist despite extensive safety testing showing no increased health risks. Many people prefer avoiding genetically modified products, driving demand for labeling and non GM alternatives. Ethical questions arise about humanity's right to alter genetic codes fundamentally. Should we modify organisms for our benefit? And where should we draw boundaries on this powerful technology? Next time, we'll explore cloning, another biotechnology raising scientific and ethical questions.